Welcome everyone to today's webinar. My name is Marie Latulip and I am the Director of Science Programs at the Institute for the Advancement of Food and Nutrition Sciences, or IAFNS. And this is the first in our 2021 webinar series that is focused on diet and impact on the gut microbiome. For those of you that are not familiar with us, we are a nonprofit scientific organization that advances food safety and nutrition science for the benefit of public health. And intellectual contributions from industry, government, and academic scientists are integrated to advance science in areas of common interest. And our overall operating philosophy is that including diverse perspectives in discussions is fundamental to the development of credible science and science that benefits the entire food ecosystem. And before we start, just a quick note on logistics. The first item to note is how to open and close your control panel. And you see that orange arrow on the top. And as an attendee, you can expand or contract that panel as needed. The second point is that there is a question panel. So we encourage you to type your questions in that box as the presentation proceeds. And those questions, they're only visible to the staff and not to the other attendees. After the presentation, we'll go back to those questions and have a dialogue with the presenter. And with that, we are very pleased to welcome Dr. Jeroen Raes. Dr. Raes is a full professor at KU Leuven. He has a substantial track record in microbiome research and has been pioneering the analysis and integration of meta-omics data sets with environmental, clinical, and dietary data. Jeroen Raes coordinates the Flemish Gut Flora Project, which you may be familiar with. This is a large-scale microbiome-focused population cohort in Belgium. And in addition, his lab is performing multiple disease-related projects, and those cover IBD, IBS, cancer, depression, and other conditions. And he develops novel approaches and tools for microbiome research. So with that, I am pleased to turn the platform over to you, Dr. Raes.
Thank you, Marie. Thank you very much for the kind invitation. Let's see if we can get this to work. I hope that everybody can see my screen here. All right. So the title of my talk is Quantitative Microbiome Profiling Population Microbiomics. It's a lot of buzzwords, but we'll come to each single one separately. I just wanted to start off with a little bit of context on my presentation. So let me take you back to the ultimate goal or promise of the microbiome field, namely that if we believe the newspapers, we're going to be diagnosing and curing all major diseases on this planet. And of course, this is a little bit of hyperbole, but the interesting part of this is that if looking out 10+ years into microbiome research, we do see a wide range of applications and disease areas being touched by the microbiome in many different ways. Sometimes it's just small-scale association studies. Sometimes there's even evidence for causality and therapy development initiated. Now, let me take you back about 10 years in time or a bit less. Because the field was in full explosions back then, and we just like many others were rushing forward into many of these clinical studies in those days, relatively small scale, to find these associations or these links between many diseases and the microbiome. Now, but at the same time, we felt a little bit awkward because we knew a lot about disease, but actually we knew very little about health. This is a very old slide, and the numbers here are not correct anymore. But it does reflect the feeling at that stage, where there was plenty of activity in the clinical areas, but a lack of knowledge on healthy variation, as opposed to, for example, in the field of genetics, where 10,000s or even 100,000s of individuals were being profiled on a regular basis. So in that area, we had a lot of backdrop in the microbiome field. We did not, but of course, it's very important to know what healthy really means if you are looking into modulating the microbiota or developing novel diagnostics. You need to know your error bars. You need to know in what direction you want to be pushing the microbiota. And so knowledge of health is just as important as knowledge of disease when developing novel products. So in that context, we started a project called the Flemish Gut Flora Project, which is a population-based study in Flanders. That's the northern side of Belgium, where we profiled a bit more than 3,000 individuals with a normal healthy microbiota and normal health situation to understand what that natural variation would look like, and especially what the confounders of microbiota research are. The first publication that came out of this work was published in 2016, and in that publication we focused on the identification of factors that could explain microbiota variation, so host factors. And we identified 69 factors in that particular study, many of which were replicated in our study, but then also were replicated in additional studies across the world. And this list, or together with other confounders that were discovered in the meantime, we feel as a bit of the gold standard of things that need to be taken into account when you're doing clinical microbiome studies and trying to assess the microbiota variation, because many of these things are also factors that pop up in patient populations. Now, I can't go through the whole list, and I invite you to go to the publication, but the two things I would like to highlight. So the single most important variable that came out of our analysis, and that is something that has been consistently coming back and back at us in every single study that we've been doing until now, is transit time. So transit time, back then we measured it as Bristol stool scale. These days we measure it as water content, is the most important confounder in microbiota research, I would say. It has effects on biodiversity, it has effects on enterotype, on composition, etc., etc. The reason why it's so important was for a while enigmatic for us. We've developed in the meantime we've developed a theory which is explained in this very nice paper by Gwen Faloni and Sarah Vera Silva, Nature Micro 2018, where we link transit time to the stage at which the microbiota is in, because we consider it as a developing ecosystem when a food bolus goes through the intestinal tract. So if you have a fast transit time, you would be, when sampling, when analyzing, you would be catching that microbiota at an early stage of development, and when transit time is slow, you would be catching it at a later stage of development. And that stage of development is on the axis going from an initial saccharolytic fermentation to a more proteolytic fermentation when the carbohydrates are being depleted. And so that is something that it plays an important role in many clinical studies, because many diseases come with phenotypes that are linked to transit time. Taking, for example, diarrhea in IBD, or for example, constipation in Parkinson's, just to name a few. So that is a very, very important factor, and I think everybody should be taking that into account somehow in clinical studies. Another factor that we feel is very important is drugs. As a category, drugs was the biggest category of factors that we had identified, and obviously things like antibiotics and laxatives pop up immediately when you think of that. But we also had other associations linked with immunosuppressants, hormones, and in other studies things like metformin and proton pump inhibitors. I mean, the drug associations are pretty impressive, and that list is getting longer and longer as we speak. What we what we also found very nice was that the initial observation that we made in our human cohort, these were associations, and we didn't know whether these effects were direct or indirect. But interestingly, a later study performed at EMBL looked at the direct effects of various drugs on gut microbiota members and replicated some of the findings that we had discovered earlier. Okay, so but after that we took things further, and in the Flemish Gut Flora Project, I mean, this is a project that's ongoing. It didn't stop at that first site, and so one of the things that puzzled us when we did the confounder analysis is that we could only explain 7 or 8% of the variation based on the extensive metadata that we had collected, ranging from blood analytes to a gazillion questionnaires. And so we thought, well, maybe host genetics is part of the missing 90+% of variation. And therefore, we performed together with the lab of Nick Timpson and Andy Franke, we performed a genome-wide association study in which we looked for loci in the human microbiota, loci in the human genome that could explain the variation of the human gut microbiota. In a nutshell, yes, we found those loci. We found 2 loci reaching the magical study level significance threshold, which is necessary in such genetic studies, and another 11 loci which reached the genome-wide significance threshold. And so again that list is in the paper, and I don't have time to go very deep on this, but one of the interesting parts was that when doing this, we worked with 2,000 something and replicated in another 1,500. Now we felt like, okay, we're just reaching the threshold. We find some loci. There probably more, but we're underpowered. And at the same time, we were also part of a large consortium called the MyBiogen consortium, in which several cohorts worldwide teamed up to reach the necessary power, or at least that's what we thought, necessary power to find a lot more loci and a lot more evidence for genome microbiome associations. And the interesting part was that in that large-scale study where we had over 20,000 individuals, we did find a few loci, but we didn't get much more. And the reason for that is probably because of the heterogeneity. The heterogeneity in almost everything, in the primers that are used for 16S, in the DNA extraction methods, in the storage protocols, in the sampling protocols, etc., etc. There's so many reasons, but maybe as opposed to other GWASs in microbiome field, because of the sensitivity of the phenotype, in this case the abundance of the organisms, at least what we read out, because of that sensitivity, the heterogeneity is a much bigger problem, it seems, than for other GWASs. So I think there's still a lot of work that needs to be done in this area of the field, where we have to spend a lot more time, I think, in standardizing and streamlining multi-cohort studies in order to get to the strong replicating results and have the power that we need to have. Okay, so I think this is still ongoing, but the frustrating part was it didn't really help solving our problem of missing variability, because maybe we added 1 or 2% of variation explained, but that was it. So that the word is still out. What are the drivers behind that, let's say, 90% of variability that we currently cannot explain? We also looked at well-being, because we felt that was also an interesting part. So in Mireia, PhD student at the time in my lab, performed an association study in the Flemish Gut Flora Project with the RAND questionnaire, looking for association between the abundance of certain organisms and parameters that describe physical and mental well-being. And so she found a few associations. You see the organisms on the right here, and that was interesting. So we found an association, but it's just one association in one cohort. And so, in order to substantiate that a bit, we looked at the RAND questionnaire, and besides the RAND questionnaire, we also looked at, for example, the diagnosis of depression, because we have medical records in our Flemish Gut Flora Project as well. And so there was some overlap. For example, the Coprococcus signal replicated between lower well-being parameters and, for example, diagnosis of depression. But we also wanted to replicate this independently. So we also we talked to our friends in Groningen, and they were so nice to see whether they could replicate some of our signals. And lo and behold, it did, right? So in the Groningen cohort, the readout was self-reported depression, and for these organisms we found this replication. And then finally we added another layer of replication on top of that, and that was a small cohort in the hospital at our site in Leuven, where we looked at the gut microbiota of severely depressed individuals that were recalcitrant to therapy, so the real extreme cases. And also there we could replicate these organisms. So this is an association study which finds a link between depression or lowered mental well-being and the microbiota, and it's nothing more than an association. However, it is one that is inhuman in a large cohort, and it replicates across multiple cohorts. And I think that is still important, an important step to progress the field of gut-brain axis research. And we're moving now forward with these organisms into functional studies. Now, you could also think about what are the reasons behind these associations. So one thing here is which is interesting is that we're talking about many of these organisms on this list are butyrate producers. So that is one link where if people that we know that butyrate has anti-inflammatory properties, and therefore people that are depressed and have low levels of these organisms, they could have a lower butyrate, and this could be then linked to neuroinflammation. Now I'm doing a whole lot of hand waving here because I have very little proof that this is the mechanism, but it is a potential interpretation of these results. Another interpretation of an association between the gut microbiota and depression could also lie not in inflammation or the neuroinflammation axis, but in the production of certain metabolites by the gut microbiota. And to study this, Mireia also did a bit of genome mining, well, not a bit, a lot of genome mining on representative gut bacteria genomes to see what their neuroactive potential was. And so basically, it was metabolic pathway mining with a set of hand-curated metabolic modules in which we looked for the potential of gut bacteria to produce and or metabolize neuroactive compounds. So things like neurotransmitters, dopamine, serotonin, GABA, and whatnot. And so the surprising result was that in this study was that a large majority of gut organisms are able to produce and or metabolize such compounds, and therefore this would also constitute a mechanism by which the microbiota interacts with our neuronal system. Now, again, lots of hand waving, lots of prediction, but no, at this point there is no proof yet, and I think a lot of the work will still need to come from intervention studies, causality studies, animal studies, etc., etc. But again, I think it's an interesting basis to start from. a lot of my talk is and will be about the bacterial fraction, but I'd like to stop for 1 second on by saying that also non-bacterial members of the gut microbiota are interesting and relevant. And in this particular case, the protist eukaryotes are, I think, also very interesting. And so this is a study done by Raul Tito, looking at the blastocystis in the Flemish Gut Flora Project, where blastocystis is generally considered a pathogen, and if you're positive for blastocystis, it means you're in trouble. And so the surprising result was, if we looked in the population, we found that 30+% were, based on the microbiome analysis, 30% of the Flemish Gut Flora Project people were positive, or they had blastocystis. That's a lot. So we wouldn't expect for that many people to be really infected by a pathogen. And then we digged a bit deeper, and indeed the story became more interesting, because blastocystis has a great number of subtypes or substrains, and a lot of those are not parasitic at all. And therefore we actually saw that the majority of blastocystis carriers are people that are carrying, in our view, perfectly benign commensal variants of blastocystis, and that the presence of these is more associated to health than anything else. So I think the message of this study is that A, the eukaryotes are important, and we should not overlook them, and B, we should not judge too quickly when we call something a pathogen, because there's a lot of variation in there, and I think the level of resolution at which we're looking at these things is very important. Okay, so for the main center of the talk, some years ago, Gwen Faloni of the lab started a project where he looked at looked into methods to develop quantitative microbiota profiles. Now, after going to a whole lot of different options with spiking and we ended up with this method, where we combine the sequencing with flow cytometry based cell counting to turn the classic profiles which you see on the left in panel A, which is a relative abundance profile. We turn that into a profile of quantitative profiles, where we measure the abundance of the organisms not in relative amounts, but in absolute, in number of cells per gram of feces. The importance of this cannot be underestimated, I think. So if you would take, for example, the panel A on the left, you would take any organism where you see 20% or 30% of that microbiota is consisting of a certain group, for example, the Bacteroides. And if you would take a profile which is on the far right of that panel A, you would also probably find one where you're also looking at 20 or 30%, whatever. It doesn't really matter what the example is, but if you just look for two things that are quite similar on the left and the right, you'll be okay. And then if you go to panel B, which is the absolute quantification, you see that if you want to interpret that right, it's going to be completely different, because something which is almost equal in relative terms can in absolute terms be completely different. That same person that had the same relative amount of Bacteroides on the left has more Bacteroides on the left, but on the right it is actually a very, very small amount in absolute terms. And when you think of this in terms of pathomechanisms, if you think of this in terms of functionality, it's actually much more relevant, I feel, to work with absolute quantifications, because the body doesn't really care about relative amounts. It cares about the amount of metabolite that is produced. It cares about the amount of immune signaling that has been happening, and that is purely linked to quantitative data to the absolute abundance of the organism. So from my perspective, I think quantitative profiling is the only way forward to better understand mechanisms in this field. You can use relative data for diagnostics, I think, but if you want to understand what's really going on, I think quantitative is essential. Now, you don't have to take my word for it, and I'll just show you some examples. So, for example, we applied this technique to a set of samples from patients, and in this particular case, we looked at the patients from a Crohn's disease cohort. And so, if you see on the left, you see the relative abundance of Bacteroides, and you see that in Crohn's disease, in CD patients, you see that they're increased when you're looking at relative data. Now, when we look at that same pattern using the absolute amounts, you see that there's no difference whatsoever. So you might wrongly conclude that the increase of Bacteroides is something that is relevant for Crohn's disease, and you start to think of all kinds of pathomechanisms, but it might not be the case, because if you look at it in cells per gram of feces, Crohn's disease patients and healthy controls have exactly the same amount of Bacteroides. And I think this is the reason why it's so important that to do these types of things, because there's no point in looking at these relative amounts when we're trying to understand how the microbiota contributes to an inflammatory disease such as Crohn's. Another example, so I've been working with the enterotypes for a very long time, since we discovered them in the early MetaHit days. The enterotypes is a way of stratifying microbiota profiles into types, and these types are described by their most dominant genera. So you have a Bacteroides type, an Ruminococcus type, and a Prevotella type. Originally, there were 3 enterotypes that were discovered, but there was other techniques that were developed for the clustering that led to these enterotypes, and one of these techniques was Dirichlet multinomial modeling, developed by Chris Quince. And when we applied Dirichlet multinomial modeling on our cohorts, we often got 4 enterotypes, and that was something very frustrating, because that was the only technique that really consistently gave 4. And we didn't really know how to interpret that until we started to placing this into the context of quantitative data, and we found actually that this 4th enterotype was indeed real and was indeed different from the other enterotypes that we had discovered, and it was actually quite special because it had a markedly lower bacterial density. So you can get up to an order of magnitude less cells per gram of feces in that 4th enterotype, which we call the B2, the Bacteroides 2 enterotype, because it's split off of the Bacteroides enterotype. And then we started mapping the patient data on that again, and surprisingly, almost all of our Crohn's disease patients in our cohort mapped on that B2 enterotype. So we suddenly had discovered or rediscovered a new enterotype that had a markedly lower microbial load, and it was associated to disease. So, and then we took it further, and then, for example, we went back to our depression cohort, and surprise, surprise, when we looked into that study, from the perspective of the enterotypes, we found that in the Flemish Gut Flora Project, people that had a B2 enterotype had a markedly lower quality of life than others. When we looked into the distribution of enterotypes over depression, we found, or over the depression phenotype, we found that in the depressed individuals, we saw a significant increase in the percentage of B2 individuals in those individuals. So somehow the B2 enterotype, the Bacteroides 2 enterotype, was linked to both having Crohn's, and it was linked to lower quality of life and depression, which was quite surprising. And we went further, and we started applying our techniques to a lot of different cohorts that we had to our disposition. And so we looked into in primary sclerosing cholangitis, PSC, in ulcerative colitis, in Crohn's, in mixed forms of PSC and IBD. And we found in almost all of these inflammatory diseases, we found increased prevalence of the B2 enterotype, where you see that, for example, for PSC, clean PSC, it's 60%, but it goes up to 80% in Crohn's disease, which was really which are really shockingly high amounts. We also looked, for example, in multiple sclerosis. Now this is still a small cohort, but we also found that certain subtypes in multiple sclerosis are also associated with having an increased prevalence of B2. And these are what the clinicians describe to me as the more inflammatory patients. And so I'm building up towards the interpretation that I want to share with you, is that when we then look at what are the common properties, what are the properties that are linked to the B2 enterotype, one of the things that we find is that in the cohorts that we looked here, we find that the individuals that have a B2, they have a higher moisture content, so their transit time is faster. And also we find their inflammatory markers to be higher. Both fecal calprotectins, which is intestinal inflammation, as well as systemic inflammation as measured by the C-reactive protein. Now, the strength of this association is quite amazing, because if we use these two parameters alone, we can actually almost predict who has what enterotype, and especially who has the B2 enterotype. So we feel that is quite strong, and of course it makes sense, right? Because this inflammatory, especially this inflammatory component, is something that can link all of the observations in the various diseases, because in IBD, yeah, that there it's obvious, but also, for example, in depression, we can think of depression as a neuroinflammatory disease. So I find this extremely interesting, and we're still applying these approaches now in a lot more cohorts and understanding this better. Now, one thing that this also says to me is that the microbiota association is not necessarily with a disease. It might actually be with a phenotype rather than a disease. And to further clarify that, I'd like to also go into some of the specific markers that we developed for some of these diseases. So some years back, we published a study on that PSC cohort, where we tried to build a signature for PSC. And so we identified a number of organisms whose abundance was associated to primary sclerosing cholangitis, and we had our signature. Now, when we revisited that with the quantitative look and with the view of taking all confounders into account, what was interesting was that we could disentangle, piece apart that signature that we had discovered before by and link it to specific organisms. Let me make this a bit more clear. Of the bacteria that were in that signature, we found, for example, some of them to be specifically associated to inflammation. For example, Fusobacteria and Veronella. And we found other bacteria to be more associated to the bile duct obstruction, which is typical for PSC, and for example, Enterococcus. And so the combined signature that we had developed is actually combined signal of various different associations with subphenotypes of the disease primary sclerosing cholangitis. And I think that's an important message. And that alone is already quite interesting, but it actually things became even more complicated when we started zooming in and combining it then with disease diagnosis. Because, for example, Veillonella and Fusobacterium are associated to inflammation, but we see that, for example, in PSC, it is that association is actually more stronger with Veillonella, while, for example, in Crohn's disease, we find the association mostly linked to Fusobacterium. So you have something which is phenotype associated, but differential in different diseases. So it gets quite complicated and it gets quite hairy, but but nevertheless also extremely interesting. So what we've done here so far was mostly from a diagnostic perspective, in the sense that what are the signals that are describing a disease or disease parameters, and can we say something about the microbiota as a diagnostic tool. But obviously we also want to go to interventions, and we want to find treatments or microbiota modulators to do so. And so I'm going to share you two areas. The first area is the area of small molecules. And so in one disease B2 association, which I haven't mentioned yet, is something that we published last summer, where is the area of metabolic syndrome and obesity associated pathologies such as heart failure and things like that. So in that progression of somebody with increasingly higher BMI, in that pre-phase of towards diabetes and metabolic syndrome, we see that the B2 enterotype also increases in prevalence. So you see that in the top left, the panel C, you see that with higher BMI, you see that proportion of individuals with B2 enterotype increasing as we go up. And so that's one very interesting observation. But it also makes sense, because we know that with increasing BMI, your transit time accelerates, and we also know that with increasing BMI, the inflammatory parameters of these individuals go up, and we indeed confirm these observations also in this cohort. And so that's quite interesting. But the even more interesting observation that was done here was when we started looking at the medication. And so if you look at the top left here, so we see that again, with going from low to high BMI, we see an increase in the prevalence of Bacteroides 2. But if we look at them, of those people that are which person that are on statins, which are the cholesterol lowering drugs, we see that people that are on statins, they don't follow this trajectory. So you can have a person with a very high BMI who was supposed to have a B2 enterotype, but they're not, right? And the only difference is that these people are on statins. So we feel that that is actually a very interesting observation. So it could mean, again, this is observational, so we don't have the ultimate causal proof of this yet, but it could mean that statin is protective of developing the B2 enterotype, the microbiome dysbiosis, which is typical for people with higher BMI on that trajectory towards metabolic syndrome. And so we think that statins might be a potential microbiota modulator. And we're also taking that forward in clinical trials as we speak. So the interesting observation I find here is that from a cross-sectional study, you can actually identify novel targets for microbiota modulation. And so this paper is the first of a series of papers that are published in the context of a European project, which is called Metacardis, and which focuses more also on other coronary issues. Now, another area of intervention is not only using small molecules, but the organisms themselves. The most brute force of all of these interventions is fecal microbiota transplantation. All well known. We're active in two areas mainly at the moment. One is ulcerative colitis, collaboration with Severin Vermeire, where we see that we get a success rate of about 20 to 30% with fecal microbiota transplantation in ulcerative colitis, which is not the almost 100% you get in Clostridium, but it's almost just as good as many of the biologicals out there. So I think it's still worthwhile pursuing. Another area where we've recently published together with Dani Deleuze from the University of Ghent is in irritable bowel syndrome, where we see indeed a significant improvement of the complaints and the phenotypes of people with IBS upon fecal transplantation. Now, this is on its own interesting, but for us it is actually the first step, because that we want to use these experiments to better understand what's going on and identify those organisms that are important for the success of such treatment, so that we can go from these bags of fecal matter to designer probiotics of the next generation, which containing organisms not from those that come from the dairy industry, but organisms that are sourced from the human body itself. Now, in order to do that, there's a few hurdles that need to be taken, and so one of the interesting things that came up from an analysis that we did in the framework of this review is that we start to know quite a bit about the gut microbiota. We start to know, if you're looking in terms of sequencing, we're catching quite a lot of the biodiversity already. But then if we start to look for of the organisms for which we know that they're there, or we have a sequence from them, we have how many of those do we actually have the organisms themselves in pure cultures? And then we're looking at an extremely small fraction, and maybe only 5% of all of the biodiversity that is out there we actually have in pure culture. And so that is something that is extremely important to mitigate. And so that's also something that we've been working on and investigating, building an internal culture collection of organisms, and we're now at more than 3,000 isolates in pure culture, which we now use for a lot of experiments and studies in anaerobic conditions, obviously. Now, one of the things that we're after is the idea that if you want to understand how the microbiota works, you need to play with it. You need to build synthetic communities in order to assess how species interact and how they crossfeed, and whether the one can suppress the other or supplant the other in mixtures, etc., etc. And I think in this area of developing designer cocktails of microbiota members, I think it's very important to also do a lot of fundamental research on how these species interact and how stable cocktails of bacteria can be made and sustained. And so this is an example that we published a couple of years back, and we still have a lot of work currently in the pipeline. And then the last step, for such cocktails of novel probiotics is, of course, these organisms are not food grade organisms, right? So they are human sourced, but they need to undergo safety studies. And so here, this is a paper which was led by Christine Verbeke, in which we then, for example, used the Flemish Gut Flora Project as a reference on an intervention study with a novel probiotic, in this particular case, Butyricoccus pylori, where we looked at the effect of administering this in healthy volunteers and to see whether they had important safety issues. In this particular case, we could see from the spread on the Flemish Gut Flora Project that there was no strong deviation of the gut microbiota upon administration. Obviously, I meant Butyrococcus pili and not pylori earlier on. And then in this whole story, I think there's an important part for the middle section, the in vitro work, and this is an opinion that we published also last year, where we say, like, well, we've done a lot of omics, and omics is really important, but I think we need to do a lot more microbiology, and especially in vitro ecology. If we want to develop modulation strategies, we need to do large-scale screenings. We will need to do modulations in different types of microbiota background, and I think in vitro work is the only area where that is possible. And of course, this should be linked to in vivo studies and then brought to human. But I think the real heavy lifting is probably going to happen in the synthetic ecology area. But this is obviously a personal opinion. So with that, I'd like to conclude. So I think to take home, I think it's important to remember that understanding normal is essential. Understanding the confounders is essential if we want to build robust diagnostics. To be honest, there's still a lot of variation we still cannot explain. We feel that quantitative profiling is extremely important for better understanding of pathomechanisms. I'm not saying that you should be all using our technique. I don't care which technique you use, but I think the world would be a better place if everybody would switch to quantitative approaches. The B2 enterotype dysbiosis, I think, is very important. We see it across many diseases, and I think it's a very important target for modulation strategies. And I think we really need to spend a lot more time on the ecology of the system and do a lot more in vitro work to understand the wiring of the gut microbiota. So I wouldn't be here without the help and the hard work of all of these guys here. On the left top, you see all of the people that were in the lab, were and are in the lab, and on the bottom right, you see a selection of many collaborators throughout the years, and funders and industrial collaborators as just as important to pay the bills of our research. And with that, I'd like to thank you for your attention, and if there's any questions, I'd be happy to answer them.